Okay, let's move on to Q&A. So in addition to our panelists and Dr. Monath, we're also joined by Dr. Scott Stibitz, right? Oh, there he is, from CBER, and who will be participating in the discussion. So there's a couple of questions online, and I guess this is probably for Lisa. So how can you assure that there'll be no mutations in the CPS knockout? How can you assure constancy over time and without, as we say in the polio trade, seroreversion?
I'm not positive that I'm the best person to answer that question. Mark, would you like to address that at all?
No. He's right there.
So the CPS1 mutant is a 6 kilobase gene deletion, so it's not going to be a reversion at the site. And we've put millions and millions of spores into multiple animals and have seen no evidence of reversion to virulence to date.
Good. We'll consider that one answered then. Thank you.
Thank you, Mark. I appreciate you taking that one on.
For Chung Yu, there's studies showing that humoral immunity does play a role in the protection provided by the FKS vaccine in mice. Should we include vaccine strategies to target both cellular and humoral arms adaptive immunity? That was one of the slides I made you skip, I'm sure.
Well, for the RCPA1 antigen, we do measure antibody production, but the antibody titer is quite low and it doesn't really show correlations. So we still have trouble to conclude whether the role of antibody is important for protection. And therefore, we focus on T cells.
Okay. Fair answer. Questions that exhaust the answers from the questions from online, or questions from the audience?
Could I ask a question?
You may indeed. Please, Scott.
So I'm very interested in the brief discussion there about the genetic attenuation. Are there other genes that you could knock out to provide further assurance for? I mean, obviously, I agree with you. It's not going to revert at the site of the mutation, but you know, it's always possible you can get suppressor mutations and so on and so forth. Your experiments in mice obviously speak to that, but I'm just curious, are there other genetic loci that could be engineered out to further attenuate?
We think there probably are, but we have not explored those in detail to this point. I think we could do so.
Fair enough.
Dr. Perlman?
Yeah, Chung Yu, I'd like to give you a chance to take a couple more minutes and say the human response that you showed in like 3 seconds. Could you maybe tell us a little more? I also have another question.
Well, we have the vaccine construct, but definitely we cannot go to immunize mice at this point yet. But then how can we assay the T cell response? So we recruit patient and also healthy subject, and then isolate their PBMC, isolate their CD4 T cells, store it, and then the rest of cell will differentiate into macrophage. And then we mix them together and then expose them to the vaccines. Remove the vaccine and then like a vaccination in Petri dish. And then wait for a couple of days until the T cell is expanded. Then we can recall with the antigen. And the data show that the patient responds to our antigen very well. And if we use healthy patients and then stimulate a couple of times and recall again, then we can see the response to our antigen as well. So right now the RCPA1 looks like pretty promising, even though we're using a heterogeneous HLA population, humans.
That's great. Congratulations. Other questions a bit broader. We really have heard today from really almost two schools of thought, and that is those using the live attenuated whole organisms, which, as Dr. Mana said, is self-adjuvanting. And then we're talking about the either recombinant proteins that Chung Yu has, or nucleic acid-based vaccines that we heard from Dr. Fuller. I wonder if we could have a discussion on the pros and cons of both, and Donna, which do you like best, since you're you're sitting on the cash there?
Well, let me try and orchestrate this, because that was the question I was essentially going to ask if pressed into making up my own question.
George, can I make a comment first? I'm Ed Robb with the Antivirus Life Science Group.
Yeah,
In
please.
addition to the efficacious work that Lisa really described, there's extensive other safety data which we have not, used for this short 50-minute presentation. But in dogs, we'll have extensive reversion to virulence and shed spread studies. And then once all that will be published in the Federal Register for public comment, hopefully by the end of 2023. And then also we'll be getting extensive field testing in client on animals in the endemic region and non-endemic region. We hope to have about 3,000 clients on animals, various breeds, different ages, different sizes of dogs. And so that whole ability to test safety in dogs will be really revealing, and really maybe try to address some of your concerns about reversion to virulence or shed spread, or other concerns about a live vaccine. So, if we can show safety on one end of the leash, we think we can show it on the other as well, right? And then proving that, dogs can be man's best friend.
I have to say, I've never heard the phrase "on one end of the leash, we can show it on the other." Maybe Deborah, could you talk a little bit about kind of pros and cons of mRNA, DNA vaccines versus subunit vaccines versus attenuated vaccines?
Yeah, sure. And I think of live attenuated vaccines, often it's really, we don't really know what are the immune responses that are providing that protection. Is you're putting pretty much the whole organism in there. And there's something in there providing protection, but is it necessary to have the whole organism? I think what nucleic acid vaccines get at is that they are, in fact, very close to live attenuated vaccines in the sense that they are expressed directly in the host. And in many respects, they do mimic a live attenuated vaccine, but what they do offer the opportunity to do is actually focus those responses on specific types of antigens or immune responses that are actually precisely needed for protection, while avoiding other sorts of immune responses that may not be advantageous, including, the potential for causing reactogenicity. The big gap there is that, with the live attenuated vaccine, if it protects, a lot of times we don't. Historically, if you look at vaccine development, a lot of times we didn't know why they protect it. It was only later. And often with live attenuated vaccine, that understanding of how it protects doesn't come till later. Nucleic acid vaccines do the reverse. We try to actually incorporate within them and figure out what is going to provide protection and incorporate only within those vaccines what's going to provide that particular protection. In many respects, this is very similar to recombinant protein vaccines because they are focusing those vaccines on those particular types of responses. I would only say that the difference between recombinant protein and nucleic acid vaccines is that you get that intracellular, in the host, direct expression of those proteins in their native conformation, similar to what you're going to get with a live attenuated vaccine. And particularly with the RNA vaccines and the self-amplifying ones, they actually are incorporating viral machinery in there that are self-adjuvanting. So they actually have that particular advantage as well. And I think overall, if you can define the immune responses needed for protection, define what antigens are going to be able to induce those responses, a nucleic acid vaccine is going to be a much better approach to go than a live attenuated one. But it's a big if, so.
Could I add a quick comment on that? If you can.
I'll say yes, Tom.
Okay, yeah, please, go ahead. I just couldn't tell who was talking.
Yeah, I was Tom Moneth. Well, I think one thing we haven't really discussed that's so critical with a vaccine against this disease is good immunological memory. We're not going to have good markers of when to have to boost. We need a durable vaccine and memory responses. So I think nucleic acid mRNA has not really proven to give durable immunity. Live vaccines all do for the most part, and they induce really good memory. So I think that's something we really need to address Clearly, in any vaccine development, memory is going to be critical.
Yeah, can I respond to that? Actually, nucleic acid vaccines are very effective in memory. What you're thinking about is a COVID-19 vaccine where antibody responses wane, and now you're not getting as good a protection. The issue there isn't that there isn't memory. There's a lot of memory there. What's the issue is that COVID-19 causes disease very, very fast, and it's not enough time for the memory response to recall and come up and shut that down as quickly as it is if it's already present. In a disease like valley fever, where it takes a little bit longer for the disease to manifest, that's that window of time that allows that memory recall response to come up. In all of the studies I've been doing for 30 years with DNA, RNA vaccines, we have very robust central memory T cells and B cells that are induced by those vaccine strategies. So it's not that they don't induce good memory, they do.
Can I add one more comment?
Please.
Okay. So for our
And
experience
in this
with,
corner, yeah, go ahead.
well, for our experience with the recombinant antigens, we can immunize mice and then wait for about 6 months. This is a pretty long period of time in the age of mice. We still can detect T cell memories in those vaccinated mice, and they are still protected.
I think the debate on vaccine modality and strategy is great, but right now we have patients that are waiting. There's people in this room that had valley fever. Most of us in this room have not. And to wait 10 years for the perfect vaccine to be designed and executed, I think is a privilege we don't really have. And so, I mean, there could be more than one strategy. Why are we thinking just one way, right? And if you really think about what we saw about climate change, what we hear from the patient advocates, what we hear from the prison systems, the debating vaccine strategy when you have something that looks pretty promising is, I think pretty selfish.
Can I ask? Go ahead. Did you want to respond?
I just wanted to say one thing. In 15 minutes, it wasn't really possible to present all of the data from that dog immunogenicity portion of the study. But I told you that only 30 dogs went to CSU, and the other 4 were necropsied on day 42. And I just wanted to say that only 1 of the 7 injection sites had any fungal growth at all, and we had to work pretty hard to find it. And there was no fungal growth detected in the draining lymph nodes from the subcutaneous vaccination sites. So while Scott's question about whether or not we could knock out a second gene to make this safer and hedge our bets, and a dog is not a human, I do think that we're a long way towards a safe product with this Delta CPS1 vaccine as it is now, and that considering that modifying this a little bit with as far along as we've gone, and for a small market disease, that we may be talking about something that's financially feasible here, as well as effective and safe with the modified live vaccine, even while we're waiting for these more technical approaches to possibly reach a point where they're ready for a cash infusion to take this to market. And this one's closer in terms of helping people right now.
Well, let me, there's a new question that's come up on the chat. And this is probably for you, Lisa, or perhaps Dr. Mana. For a vaccine trial, and I'm characterizing this as the Dalmatian question, right? For vaccine trials in dogs, is there consideration to immunize dogs who have weakened immune responses or breeds that are known to be more susceptible?
Yes, we'll vaccinate them all. I don't know how we're going to detect whether they have weakened immune responses because we don't have very good T cell assays in dogs. And we're not going to know, but we're not going to select dogs based on, this breed does really well with it. We're only going to vaccinate beagles.
Well, how about breeds that don't do well? Are you going to oversample them?
We're not going to oversample anything.
Okay.
I mean, I think that would be the wrong approach, actually. And I can't tell you how to tell whether a dog is immunocompromised unless he, comes in on a milligram per kilogram per day of immunoglobulin, or maybe he's been taking a JAK1 inhibitor for 2 years.
But in terms of breeds, though, right?
There are some breed susceptibilities, but we wouldn't either overrepresent them or underrepresent them in a safety study.
Dr. Mana, do you have comments on that?
Yeah, thank you. I think obviously in the clinical trial program, you would exclude people with known immunodeficiencies or on immunosuppressive drugs. And then as the vaccine was used, perhaps, an experience would accumulate where that would indicate safety in people with immunodeficiency, just because some of them will get vaccinated and hopefully a body of data will be. So many live vaccines are safe in immunocompromised people. There are few not. A smallpox vaccine is an example. But I think there's a reasonable, just because of the mechanism of attenuation here, which probably doesn't depend on the immune system, but rather the genetic defect in the vaccine. I've already seen in mouse models that it's safe. So I would expect that will be the case. Whether it will be sufficiently immunogenic to protect people with immunodeficiencies, hard to say. But I think safety is likely to be acceptable.
Thank you.
We had
Dr. Ample?
Yeah, thanks, George. This may be directed toward Donna, but all of you. The one thing I haven't heard here, and it should be part of the plan, is vaccine uptake. That's, of course, a huge issue. We saw it with COVID, and we saw vaccine, the original Merck Lyme vaccine, because of resistance among people. So has there been any discussion? And I think that could be platform dependent or related as well. So are there any planning, if we get any of these three platforms or more, what might the population uptake be, or will it be resistance to being vaccinated in or out of the endemic area?
It's a great
Maybe I'll tackle that question. I'll tell you,
Royce,
and I don't know what
please.
John's answer to this would be, but we had an enormous trouble and I think a better time for trying to recruit people to the vaccine project. We spent a good deal of time on doing Sunday afternoon news shows, and we hired one of my 3.5 full-time employees was a woman that was very well connected in the community that ended up on the cover of People magazine. I don't know whether, I'm sure John and maybe Neil or a couple others would remember her picture on People magazine. And we were in, the vaccine project was in Time magazine. But nonetheless, we had a heck of a time recruiting. We spent 6 years recruiting less people than we had intended. So it was a lot of work to recruit. And like I said, the people that tended to sign up tended to be educated. And so the actual target population that we wanted to reach were the people that didn't sign up. You notice, so even in the 1980s, Latinx people were probably 35 or 40% of the population of Kern County, and we recruited hardly any. So recruitment to this project is a huge issue and requires a very big budget. The only way you sell Tide is by having it on television every 30 minutes.
Can I ask one more question? This is more for Donna. I'm Dave Barrett. I'm at Anavive Life Sciences. Based on the conversations we heard yesterday about therapeutics and today and about the vaccines going forward and the lack of interest, I guess, on corporate sides, large companies to take on these things. We're a nice company. I'm a nice guy. I've been rejected by every large human vaccine pharmaceutical company on the planet with respect to the vaccine. And it's not because they don't like the vaccine. It's not because they don't think the vaccine will be safe and effective. It's because they don't think they can make enough money off the vaccine. And they also say, if you come to us with funding and you can help advance the vaccine forward more than you have, and by funding, I mean $100 million in funding. I don't, I'll take whatever you write a check for, Donna. I'll take anything you have. But to advance it, as Tom was saying, you're going to need $200 to $300 million at a minimum. We really need funding from the government or from agencies like yours to help us push it forward, or else it's just going to sit.
When we were doing the vaccine, Valley Fever vaccine project, we went way down the path of romancing the military. Thinking that was a good potential source for not only for trialists, for trial candidates, but as an end-stage purchaser. it sort of fell on deaf ears, I'd have to say. It still struck me as kind of a reasonable thing to do. I don't know if you remember.
We've tried with BARDA. Very nice conversations with them, and also very nice when you have more funding, come back. So.
Yeah, you're too young to remember this. I'm just guessing. But
thank you, but no.
In 1990, at the start of the first Gulf War, all the Marine snipers were sent somewhere by Lemoore to practice their skills, and they dug in by pulling creosote bushes out of the desert and
Hunter Liggett.
Was it Hunter Liggett?
Yep.
Yeah. Anyway, he's from Monterey County originally, so he knows. But and there was something like 40% reduction in troop size because of that. We thought that might stimulate a little bit more interest, but we went way down that path, and including long trips to Fort Irwin and 29 Palms, and which were amazing experiences, I'd have to say. I could tell you stories about Fort Irwin, but they were, they kind of didn't want to hear it. They didn't want another thing on their plate. That's sort of what it boiled down to. So then that's why burden of disease studies are so important.
Yeah. I can just respond quickly. I think that it's not that we don't want to hear it. It's that our mission is to get products maybe so far down the path, and obviously $100 million might be outside the scope of what NIAID could do. And happily, somebody asked, which program do we like the best? I don't think we choose that way. Happily, we've supported or are currently supporting all of these programs, and so. But I think it is something for the community as a whole to think about. Once these programs progress so far, where is the next step coming from? And it might be outside the scope of NIH for $100 million.
So big pharma is not going to be interested in this product, but there are specialty pharmaceutical companies that might be, and we've done modeling to look at whether there is a sufficient return on investment, and concluded there was for a smaller company interested in niche products to engage. So I think, but it's got to be, as Donna said, the fire's got to be lit first, I think, by moving forward, I think, into the clinic. I think phase 1 trial with good immunological markers is really the key step if we're ever going to succeed.
Thank you.
Yeah, I just quick comment on that. For small companies that develop platform technologies, the incentive, even if it's a more of an orphan sort of disease, is a proof of concept with your platform that then can be adapted to other things. So the thinking among the biotechs I work with is that, if we're successful with this, and this is like the first fungal vaccine ever, that puts their platform on the map as a place to go for, other fungal infections. So I think it's just, it's a different sort of pitch or angle to go after.
Dr. Thompson?
Yeah, G.R. Thompson from UC Davis. It's fun to talk about the design of these trials, and I think that this is going to be borne out over the next few years. But one of the questions I've had as we think through the actual design of the study is we have to prove these patients don't have pre-existing immunity. And John touched on this a little bit in his talk, and Royce and I have talked a little bit. And, I'm struck by in tuberculosis, we skin test older patients twice, right? We test them a month apart to see if their immunologic memory gives that second test a positive. Was that ever done with the spirulin test? Or in Spherusol, in the prison study, there was a big negativity rate, but they never were tested a second time to see if they had a booster phenomenon. And I'm sort of just curious about that with Coxie skin testing. Has the booster phenomenon been looked at? Because that's going to be something important for a vaccine study.
Dr. Johnson.
Well, one of the problems with skin testing is, and John and I were talking about this, is it's very operator dependent. So you've seen all kinds of data about what the sensitivity of Spherusol is, and there's been various and sundry conjectures about why that is. I'm reasonably certain that all of that is operator dependent. So the paper that Neil wrote, but I'm the first author, I did most of this. I did most of the skin testing, however, but myself in getting Spherusol back, or getting a spherulin product that is preserved in phenol back on the market. So the problem was that spherulin went off the market because the companies lost data that the FDA demanded they keep, and it was sold like 3 times. And so Hilly Levine and eventually Stuart Nielsen and I and eventually Neil and a few other people got this stuff back. It actually is the same exact antigen as spherulin. It's not made the same way. It is the same lot that spherulin was made from. They still make spherosol from it. It's only the preservative. When I did the dose response curve down to the microgram, it was exactly the same as Hilly's original work. But what I discovered was that there were people that had false positive skin tests to the preservative because they had hypersensitivity to the mercury, which had not been noted by Hilly because it had been tested in Mexico, just north of Mexico City. And so and we got 98% sensitivity. Now, Janice recently, who's here, reported 72%. But Janice didn't do those skin tests. That was done by the lab in the Mayo Clinic. When I tested my own laboratory people about doing PPDs and Coxy serology or Coxy skin testing, I found that there was like 100% disagreement between 5 people trying to put on and read a skin test. So skin tests are highly operator dependent. If you're going to use a skin test to reject people, and I think you could use spherosol for this, you have to have expertly trained 1 or 2 individuals that are going to do it at any given site to make it a reasonable screening test.
Thank you.
The other thing we might want is, because skin testing didn't tell you were immunized against the whole spheral vaccine. That was also looked at. I don't have that data. Maybe John remembers it. But we need a surrogate to show you're immunized. I think that's extremely important. And I think maybe we've heard that we have that, but it might be expensive to do on a big scale basis.
Let me ask one last question while we still have this thing, and Kim Lucas, stick your fingers in your ears. Donna, would NIAID consider a prisoner trial for this?
You know, when I saw, that's one of our checklist questions, and I would have to check, but I actually, my supervisor's here, Sam, I don't know if you know, but I highly doubt it. I don't know. I would have to check. But it doesn't. It seems to me that there are quite a few gaps here before you're ready for a $100 million infusion. I don't know how, you all are experts, but it seems like the correlates of protection and how you're going to test this population, whatever that population is, sort of needs to be well defined before, and manufacturing has to be worked out. So, I mean, I understand that the total amount seems insurmountable, but it does seem like there's a lot of other steps between there and now that maybe NIAID could help with these gap filling issues. And there seems to be some discussion that needs to take place in the field to agree on these best next steps.
Thank you very much.
Could I make one more comment about prisoners? One, I don't think it'll ever happen. And number 2, did you look at the skin test uptake that was presented in the prisons?
12%.
Yeah. So prisoners are the worst at signing up. And nobody's going to let you do it on other than a voluntary basis. So I think that even if the FDA and the federal government agreed to do prisoners, I don't think the prisoners would agree to do prisoners.
And that's the last word. We're at 0 time.